Hello everyone, I, Ishan Shah, am going to provide you with a brief overview regarding the concept of multi-locus sequence typing, or better known as MLST. So what exactly is MLST? We all know that each gene has alternating forms or variants known as alleles. Every gene occupies a specific position or location on the chromosome, which we call as the locus. As a result, multiple genes occupy multiple positions or multiple loci. However, mutations in a gene can give rise to multiple variants and a series of alternating expressions. This concept of multiple alleles, of multiple genes that are situated on multiple loci, has been exploited in MLST. MLST is a popular genotypic method for taxonomical classification and was first proposed by Malde in the year 1998. It is based on the principle of multi-locus enzyme electrophoresis, but differs in the fact that in multi-locus enzyme electrophoresis, we are analyzing the products encoded by the genes, that is the enzymes, while here in MLST, we are directly using the genes and the necessary variants. As I just mentioned, it identifies allelic variants to categorize, subtype, and further classify members of haploid bacterial population, that is organisms which have a single set of chromosomes. It can also be used for diploid organisms in the form of high throughput MLST. Now coming to the procedure, MLST takes into account sequencing of several different housekeeping genes. Housekeeping genes are the genes that are essential for the survival, and it compares it with sequences of the same gene from different strains of the organism. For each gene, approximately 450 to 500 base pair sequence is amplified using PCR and then sequenced using Sanger's method. Here in Sanger's method, 7 housekeeping genes are sequenced one at a time. Each nucleotide along the sequence is compared and any differences are noted. Like I previously mentioned, each difference or sequence variant is called allele and is assigned a specific allelic number. Hence, each strain will have a series of numbers known as allelic profile, or in this case, sequence type. Now, if two strains have identical sequences for a particular gene, then they'll have the same allelic number for that gene. However, if the two strains have identical sequences for all the genes, they'll have the same allelic profile or sequence type. This relatedness between each allelic profile is expressed in the form of dendrograms that vary from 0 for strains that are identical to 1 in case the strains are distinctly related. So this is a pictorial representation of the protocol followed in MLST. Here is a typical prokaryotic chromosome, and these are the 7 housekeeping genes that have been taken into consideration and are placed at equal distance from each other. This is followed by amplification by PCR and Sanger sequencing. And lastly, after comparative analysis, all the genes have been assigned their allelic numbers. Coming to the applications of MLST. MLST can be used for long-term investigation of bacterial populations, especially when subtyping bacterial species which have a high rate of genetic recombination. It can also be used in clinical microbiology, where one needs to differentiate strains of a particular pathogen. For example, some strains within the species Escherichia coli K12 are completely harmless, whereas others like O157H7 can cause fatal and serious infections. Thirdly, it can also be used to track a virulent strain as it moves through populations, which forms a part of epidemiological studies. MLST is applied to determine geographic distribution of different strains, as depending on the environment, physical factors, and amount of nutrients available, the gene expression of the organism will vary. And lastly, it is used as a reference tool for comparison of isolates between laboratories or even different geographic areas. Advantages. The sequence data that is generated is unambiguous, easy to standardize, electronically portable, and hence highly comparable. It overcomes the limitation of rRNA analysis, which focuses only on a single gene. Here we are focusing on 7 genes. Allelic profiles can be obtained directly from clinical material like blood or CSF. Material that is needed for MLST, such as DNA or blood, is easily transported among the labs. Hence, there is no such processing of the sample required, and it can be extracted easily. Importantly, it has excellent resolving power to distinguish among very closely related strains. Now, what are the limitations or drawbacks of MLST? Firstly, the alleles that are assigned to the numbering system is not a representative of the actual gene sequence, which makes the phylogenetic analysis of the strains poorly credible. Sequencing of 7 genes is costly, time-consuming, and laborious, as we are only sequencing one single gene at a time. There is no single set of universal housekeeping genes that can be used for all pathogens. So for each organism, different set of genes will have to be taken into consideration. Fourth drawback of MLST is that the 7 genes represent only a tiny fraction of the data present in the whole genome. A bacterial genome typically consists of 300 to 3000 to 4000 genes, while here we are only analyzing 7 genes. Strains that are identical by MLST may be genomically different. Importantly, not all pathogens are suitable for MLST methods. Example, some pathogens like Mycobacterium tuberculosis exhibit very little variation throughout the entire genome, which proves to be insufficient in differentiating strains. Conversely, some bacterial genomes have accumulated so much variation that MLST housekeeping genes do not provide adequate information for typing. Another major drawback of MLST is it is not useful for comparing the organisms above the species level. So, who comes up with these MLST schemes? Experts in the area of particular pathogen provide the necessary MLST schemes for that organism. Here are some of the sources where MLST sequences are maintained, and these databases can be used to compare the necessary sequence. This is just a comparison of MLST with other genomic techniques. As we can see, the ease of MLST, result interpretation, and the time required for the test is not on the favorable side. However, the reproducibility between and among labs is quite good. Advances in MLST technology, high throughput MLST, which I previously mentioned. Most of the MLST data that is generated by Sanger sequencing is now replaced by high throughput technologies such as pyrosequencing, next generation sequencing, and whole genome sequencing. These technologies can accurately read lengths up to 10 kilobases and in turn generate up to 50 million pair end reads per run. Importantly, with the use of barcode primers, hundreds of genes can be sequenced at the same time, leading to reduction in cost and time duration. Importantly, it can also be used for diploid organisms, which carry 2 alleles for each gene. Next generation sequencing allows separate analysis of both the alleles, unlike Sanger sequencing, where the resulting sequence is the mixture of both the allelic sequences. Carrying on from advances in MLST, here is its expansion in terms of gene by gene approach. That is, we are combining genomics and sequence typing, through which we can recover the entire genome of an organism for analysis. Traditional MLST involved only 7 housekeeping genes, but With these hyphenated techniques, new molecular markers have been incorporated. These include RMLST, which involves variation of 53 genes that code for bacterial ribosome protein subunits. We also have core genome and whole genome MLST, which offer the highest resolution using gene by gene approach. Or we could even use meta MLST, where we combine MLST with the high throughput of metagenomics and can analyze different genomes in a bacterial community. So lastly, here is the comparison of taxonomic resolution of various molecular techniques. And we can see that traditional MLSE classifies organisms up to the strain level. Here in this flowchart, we can see that 16S rRNA sequencing, which is one of the most common methods for bacterial taxonomical classification, involves only a single locus and hence can differentiate only up to the genus level. On the other hand, with high throughput MLST techniques, we can actually study the diversity up to clone level due to sequencing of a large number of genes. So these are the references which I've used to collect and collate the data. Thank you.